Okay, so now that we understand that there is a codon language, how does it do that? How do the codons of the messenger RNA get made into the protein? And so the way that happens is you have this molecule called a transfer RNA. And the transfer RNA has two regions to it. It has an anticodon, which is the exact opposite of the codon. So remember that codon was that 3 nucleotides, 1, 2, 3 on the messenger RNA. Well, that anticodon is the complementary base pair to that. So we call it an anticodon. So A would be paired to U would be paired to A, and G would be paired to C. So on my transfer RNA or my tRNA, we have an anticodon, which is complementary base paired to the codon located on the messenger RNA. We also have on the other end an amino acid that the codon codes for. So this amino acid will be added to the polypeptide chain. So let's go ahead and look at initiation. And initiation is getting that first amino acid in place, forming the complex so that the polypeptide can be made from the messenger RNA. So the first thing that's going to happen is if we look at this, we're going to have our messenger RNA here, and it's going to bind to the small ribosomal subunit. This is the small ribosomal subunit in this complex, and it's going to bind at a messenger RNA binding site. So right here you can see there's a binding site. This messenger RNA is going to come in. It's going to bind to the binding site on the small ribosomal subunits. Then, second, what's going to happen is you're going to have your initiator transfer RNA. And remember, that initiator transfer RNA is always a methionine. It's always a methionine. That is our start codon. So that start codon is going to come in and it is going to bind to the complementary messenger RNA. And remember, our start codon, the messenger RNA says AUG. That's your mRNA. And that means that the anticodon from the tRNA is going to be UAC. So that's our anticodon. That's here, UAC. And then on top is going to be our amino acid that is associated with UAC, and that is methionine. Once this complex here is in place, then the large subunit comes in and binds to the messenger RNA. Quickly, if we look at what's involved, we saw the activation, but let's look at what's involved in the elongation. So in order to do that, you're going to look at 3 sites. We have a P site, we have a site, and an E site. So the P site is the site that holds the polypeptide in place while it's adding a new amino acid. And so it has the name peptidyl site. So your P site is your peptidyl site, or peptide site, if you want to say, to remember it. But this is your P site. This is what holds the polypeptide structure in place for the amino acid that is on your tRNA to be added to that peptide. Then we have the A site, and this is the empty site where tRNA comes in and with its amino acid, and that amino acid is going to be transferred. So it's called the A site mostly because it's called the A site because it stands for amino acyl tRNA. You do not need to know that word, but that's why it's called the A site. So you're empty, this site is empty, and a new tRNA comes in with its amino acid right here. Then the E stands for exit. And that is after your tRNA has donated its amino acid, it leaves through this site. When it leaves through the site, it goes to be activated or recharged. So, and what happens is transfer RNA comes in with its amino acid, the P site holds it in place and transfers the amino acid onto the peptide chain, and then it is shifted out and it exits the E site. And this happens over and over and over again, and that gives you the elongation. That is what is making that peptide chain. Okay. So here we have our sites again. So the ribosome has 3 binding sites. The P site that binds the transfer RNA holding the peptide chain in place. The A site that binds the tRNA holding the next amino acid that's going to be added to the P site. And then the E site from which the tRNA is discharged, it exits after it loses its amino acid. The primary role of transfer RNA is to carry the amino acids to the ribosome so that they can be added to the polypeptide chain. So that's all that it is for. It is to transfer the amino acids that are out there, and the amino acids are going to be up here. The amino acid, to transfer that amino acid to that growing peptide chain. So the two parts of the transfer RNA that we need you to know is the amino acid attachment site. So this is the top of the amino acid here at that 3' end. And then it's also the anticodon region right here. So this anticodon region, remember, it's reading a messenger RNA. This is my messenger RNA. And the anticodon is the complementary base pair in that codon, that reading frame. So in this case, C is complementary to G, U is complementary to A, C is complementary to G. So if I look at this tRNA, you can see that I have some loops here. It doesn't matter. We're not going to really talk about the loops. You don't need to know too much about these loops. And also you see that the RNA binds back on itself. So it's not in a helical form. So don't confuse it with the DNA that's in that double helix. But it does in places have this double RNA strand that you can see here. And if I follow this, this is only one segment of DNA. This is only one segment of RNA. This is not two separate strands of RNA. You have a 3' end here, and literally, if I were to open all of this up and take away the bonding, because this is just folding of it, this is all still that one RNA molecule, and then I have my 5' end here. Okay, so what happens to the transfer RNA once it leaves the ribosome, once it leaves that exit site, that E site? Well, as it turns out, that transfer RNA needs to be recharged. And there are 4 basic steps in that recharging of the transfer RNA. So to start with, you're going to have an enzyme, which you see here. This enzyme is specific for an amino acid. It's also specific for a tRNA. So you have 20 amino acids, therefore you're going to have 20 of these specific enzymes. It's very important that we mention the specificity of these enzymes when we're talking about them in the IB exam. So you have a specific enzyme that binds to a specific amino acid. Then that enzyme also binds ATP, and a dephosphorylation occurs. The phosphate leaves, which is what you see here. And you have your transfer, your specific tRNA that is specific again for the amino acid. It will now come in and bind to your enzyme. Your enzyme uses that last energy of AMP to covalently bond this amino acid to the transfer RNA. So ATP is going from ATP, ADP to AMP. So what does that tell you? That's adenosine triphosphate, adenosine diphosphate. So it used 2 phosphates in this dephosphorylation reaction here, leaving you with the adenosine monophosphate. That adenosine monophosphate still has a little bit of energy left, and that last little bit of energy is used to covalently attach your amino acid to your transfer RNA. Then your transfer RNA will go back to the ribosome and add its appropriate amino acid to the appropriate codon site. All right, so elongation. What is elongation? Remember, I told you elongation is literally where we're adding the amino acids one at a time to the polypeptide chain. So let me go. If I start here, let's go ahead and start where we have the transfer RNA carrying the amino acid cycling through the EPA sites. So we're going to start with cycling through. We'll start right here on this one. Let's put this one as 1. So you have the polypeptide chain here being held in place. You have the matching anticodon here with the amino acid here. This tRNA binds to the matching codon. The anticodon binds to the codon. And now what you have is when they're next to each other, you have a polypeptide link. So this link is forming between these 2 amino acids. Then what you have is you have a transfer. It's holding this in place, but these are linked. So it transfers, this polypeptide chain transfers onto the new amino acid. So you see that here. You have the new amino acid, this one here. Then you have the chain that started there. This is now an empty tRNA. This empty tRNA will move to the E site where it will exit and go for recharging. The elongation process is a process or a cycle that happens over and over and over again until you get to a stop codon. So first step, a transfer RNA carrying the next amino acid binds to the A site. That's what we see here. The tRNA in the P site holds the current chain. That's what it's doing. A peptide bond forms between the condensation reaction of the amino acids at the A site. So you have a condensation reaction happening here and the formation of a peptide bond. And the last, so this is now going to be the last amino acid in the chain when we see it, and here it is. So we have the peptide bond between the amino acids formed through a condensation. This is now the last amino acid in the chain. The ribosome moves down the chain 3 bases, shifting the tRNA from an A to a P site. So this one from an A site is going to go to the P site, and that's what we have here. And then this one is going to go from a P site to an E site, and that's what we have here. And P site to E site. The transfer RNA at the E site is now discharged, so this guy is gone. And the process repeats itself. This guy is leaving to be recharged, to be activated. This elongation continues until one of 3 stop codons are encountered. When the stop codons are encountered, this signals a termination factor or a release factor to come in and bind to the ribosome. When that happens, the results are disassembly of the translation components. So literally that release factor comes in here, right here, and it binds right here to the ribosome here, and of course the tRNA, the mRNA right here through the anticodon. When this is done, the peptide chain is released and the messenger RNA is degraded, and the ribosomal unit itself disassembles. Okay, so if we have a quick look at prokaryotic and eukaryotic translation, what we're seeing here is that if we look at the free ribosomes, those ribosomes are basically producing proteins that are going to be used by the cell within the cell. And if we look at our prokaryotes here, again, we only have free ribosomes. We don't have any bound ribosomes. We don't have any ribosomes bound
proteins that are bound
endoplasmic reticulum, are primarily secreted, but either they're secreted and used for lysosomes, or they're secreted outside the cell, or they are actually secreted to be used within the cell membrane itself. And in eukaryotic cells, replication and transcription take place in the nucleus while translation occurs in the cytoplasm. Because we have that nuclear membrane, we're able to compartmentalize these 2 activities. And then don't forget that before this RNA can leave the nucleus in a eukaryote, it has to be post-transcriptionally modified. If, however, we look at prokaryotic cells, there is no nucleus. So as a result, you literally have transcription and translation happening almost simultaneously. As it's being transcribed, as soon as it gets long enough, it's being translated. And prokaryotic cells do not have introns. There are no introns. Remember, eukaryotic cells, we have that post-transcriptional processing. Yep. So that post-transcriptional processing is also where we splice out the introns. So those are those intervening sequences that are not coded for. You don't have those in prokaryotes, but you do have those in eukaryotes. I hope I didn't say this wrong, but eukaryotes are where we have the post-transcriptional processing. And we splice out the introns and add the 5' cap and the 3' tail. Okay, so just briefly, transcription, translation are coupled together. In prokaryotes, the RNA is not modified. In eukaryotes, they are not processed together. You have them in 2 separate compartments, either in the nucleus or in the cytoplasm, and the RNA is modified. So realize that when we're talking about this, I don't want you guys to get the idea that we're talking about one ribosome copying one messenger RNA and then it's degraded. That's not the way it happens. We have these polysome structures, and you can see them if you look here. These are all ribosomes attached to messenger RNA. So you have lots of ribosomes making lots and lots of these copies of the protein, of the messenger RNA into protein. So we call all of when we have a whole bunch of ribosomes on a single strand of messenger RNA, we call them polysomes. So recall that in prokaryotes you have DNA, RNA, and translation of into proteins all happening at the same time. So you see your strand of DNA here, but you also see coming off of it, it's being copied into RNA. And in addition to that, you see ribosomes attached straight away. And again, you have polysomes on your protein.